Welcome to What's on the MCAT. I'm Matt, and today we're going to be doing the problem set from topic 2 in the biology and biochemistry section of the MCAT. Here we go. 1. A cell exhibits a defect in modifying, sorting, and packaging proteins and lipids for secretion or delivery to other organelles. Which organelle is most likely malfunctioning? A. Smooth endoplasmic reticulum, B. Nucleolus, C. Golgi apparatus, or D. Lysosome? And the answer here, of course, is the Golgi apparatus. This is the part of the cell where vesicles are formed and the modification, sorting, packaging, and transportation prep is done for the cells. Number 2, a drug that prevents the polymerization of actin filaments would most directly inhibit which of the following cellular processes? A. Formation of the mitotic spindle. B. Amoeboid movement and cytokinesis. C. Maintaining the shape of the nucleus. Or D. Transport of vesicles from the ER to the Golgi. Now, the actin filaments are responsible for amoeboid movement, which is kind of like cell crawling, and for cytokinesis. And this would be answer choice B. Keep in mind that this process is dynamic and involves the active polymerization of actin microfilaments in real time. Units are actually added and subtracted as this process occurs. Number 3, which of the following is a key structural difference that can be used to distinguish between bacteria and archaea? A. Presence of a cell nucleus. B. The mechanism of flagellar propulsion. C. Presence of ribosomes. Or D. Chemical composition of the cell wall. Now, you may recall that neither bacteria nor archaea have a cell nucleus, and they don't differ in B or C either. This leaves us with D, the chemical composition of the cell wall. Now, the classic thing to remember here is that bacteria are formed with walls that contain peptidoglycan. This is unique to bacteria and not found in archaea. Number 4, the life cycle of HIV, a retrovirus, is distinct from that of most other animal viruses because it involves A. Lysis of the host cell to release new viral particles, B. Synthesis of DNA from an RNA template, C. The use of host cell machinery to replicate the viral genome, or D. The self-assembly of new viral particles in the host cytoplasm. Now, you may remember that most organisms that are infected by viruses that are not retroviruses may undergo lysis of their own host cells. Their cellular machinery may be hijacked by the virus, and the virus may undergo self-directed self-assembly of the new virions in the cytoplasm of the host cell. This knocks out A, C, and D, leaving B, the synthesis of DNA from an RNA template. And of course, this is the signature of a retrovirus. Retroviruses specifically employ reverse transcriptase to turn the fundamental dogma of biology on its head and to go backwards from RNA to DNA in order to insert themselves into the host genome. Number 5, during which phase of mitosis do sister chromatids separate and begin to move towards the opposite poles of the cell? A. Telophase, B. Metaphase, C. Anaphase, or D. Prophase. Now, I have a really funny way of remembering this. If you look at an animation of what happens to chromatids as they get ripped apart from their sister chromatid and pulled towards opposite ends of the cell, it looks like a pair of arms, almost like the letter A sideways. And I picture the tiny little chromatids saying, ah, as they're pulled into their separate daughter cells. If you can remember that, ah, as they're being pulled to separate poles of the cell, you will remember A for anaphase. I encourage people to find funny, silly ways to visualize memorization of the many facts that you have to know for the MCAT, as this can make it easier to recall on test day. Number 6, a mutation in a gene responsible for a G1 checkpoint protein could most directly lead to which outcome? A, the cell entering S phase with damaged DNA. B, failure of sister chromatids to separate properly during anaphase. C, the formation of a faulty mitotic spindle. D, premature exit from mitosis before cytokinesis is complete. Now, the key here is to remember when G1 occurs. This is a checkpoint that occurs just before entering the synthesis phase of the cell cycle. This means that it's supposed to stop the cell from entering synthesis phase with damaged DNA. This is answer choice A. Of course, if G1 is working, then you won't have the later damaged DNA passed on through the remainder of cell replication. Number 7. The major structures of the nervous system, including the brain and spinal cord, arise from which of the primary germ layers during embryogenesis? A. Neural crest, B. Mesoderm, C. Endoderm, or D. Ectoderm. And the answer choice here is ectoderm. The easy way to remember this is that there is a famous saying, the ectoderm is your attractoderm. The thing that makes a person attractive as a human might be their skin, their eyes, maybe their brain that gives you their personality. And it just so much turns out, and it just so happens to turn out, that those things are the product of the ectoderm. Number 8. In a laboratory experiment, a non-pathogenic strain of Streptococcus pneumoniae is cultured in a medium containing DNA fragments extracted from a heat-killed pathogenic strain. After some time, some of the non-pathogenic bacteria are found to have become pathogenic. This phenomenon is known as A. Binary fission, B. Transduction, C. Transformation, or D. Conjugation. Now, this problem has a fascinating implication. There is a heat-killed pathogenic strain that left behind DNA fragments. When these are taken up by another strain and they obtain that DNA trait, this is called transformation. Answer choice C. Can you imagine if humans did this? If we could just absorb the DNA from someone else and take on that trait? Bacteria are pretty cool. Number 9. Which of the following transport mechanisms requires the direct hydrolysis of ATP to move a solute against its concentration gradient? A. Primary active transport, B. Facilitated diffusion, C. Secondary active transport, or D. Osmosis. Now, anytime we're considering pumping ions directly against their concentration gradient by burning ATP, we are discussing primary active transport. Answer choice A. Number 10. The developmental process by which a less specialized cell becomes a more specialized cell type is known as A. Apoptosis, B. Gastrulation, C. Determination, or D. Differentiation. And the answer here is D. Differentiation. Thank you so much for joining me for problems from Section 2 on the Bio and Biochemistry section of the MCAT. Reach out at www.mcatboss.com or mattinmedicine.com for personal MCAT help. I'm one of America's most experienced MCAT tutors, and I'm here to help be your guide and coach as you apply to, interview for, and start your medical school pathway and your career as a doctor.